אז זה כיסא מלא אבק. זה יפה. אוקיי. חד. Okay, so we are still in the very basic, early description of sensory systems, which is talking about some concepts or some basic issues that you will encounter along the way when we talk about sensory systems. I left you last time with just giving you a flavor of different ways to quantify behaviors, and I think we were here talking about D prime, which is commonly used, a commonly used measure in decision making or discriminability of how can we discriminate between two different stimuli. But there are again other methods that you will see all along the way. Each one has its advantages and disadvantages. Another very common way to quantify perceptions is the measurements of response times. And it's intuitive to understand that the more time the brain has to process information, the more accurate it can be in difficult decisions. So this is called speed-accuracy trade-off. The faster one has to sample, the sampling time is limited, the lower is accuracy in the test, and this is one example of something that when I'll teach olfaction we'll talk about. There is a discrepancy, there is an argument in the field about whether there is speed-accuracy trade-off in olfaction too. And this is just one figure from one of those papers showing classical speed-accuracy trade-off relationship, and this can be calculated to say how much time it takes for the nervous system to discriminate one order from another, one stimulus from another, and you can see that how the tasks are shown here in red, for example, in the red curves. So they will be, if you look at the same time, it will be less accurate for hard tasks, and it will take you more time to get more and more accurate. And if the task is easier for a given time, you will perform better. And this has some consequences also on the limits of accuracy that one can get. So some stimuli you cannot differentiate. So if it's hard stimuli, you will never get beyond, I don't know, 80% in this case. If these are easy tasks, you can get to 100% accuracy. Okay, so speed times is also something that is very common in quantifying perceptions. I remind you, we're still in the era of psychophysics. Okay, historically, the community who started this the whole sensory systems field is the psychophysicists and psychologists that started to quantify perceptions. And today we can also do it in experimental animals, where it's much harder to, of course, ask the subject how does he feel. So many tasks are still limited to humans that can give us good feedback about their perceptions. Okay, another flavor of looking at response times has been actually quite important in the visual field. And this can give you a flavor of what is the task at hand of the nervous system. So, for example, this is a well-known example by Ant Riesman, which is, I think, a good colleague of Shaul Hochstein, which will teach you the part of vision. I think he's actually writing a book on vision now, so I think it's still great that you can that he's teaching and you can take courses with him anyway. But so if you look at this kind of task where one has to differentiate where is the blue cross in this type of task, okay, so the task is to find the blue cross. You can have this kind of matrix, this kind of matrix, and this kind of matrix. Of course, here the blue pops out also, especially because of its color, and it's easy to find the blue cross popping out. This is a little bit more difficult because the cross is only different in the structure and not the color, and the last one is more difficult because it's combining both structure and color. So the question is, what is the strategy people use when they have to find the blue cross? And in fact, when Anne Treisman started quantifying the response times to this kind of visual task, she found that it doesn't matter how big the matrix is in the first task, the response times would stay the same. It's as easy to find the blue cross here in this kind of a task, regardless of the size of the matrix. Okay, and this means that actually perceptually we're having what she called the parallel search, and this is just some data from these kind of experiments where the subject had to give a response in a given time. In this case, it's a half a second, and it was half a second regardless of the size of the matrix. And here she asked, one second, she asked whether the cross is there a cross in the matrix or is it absent in the matrix? And in both cases, it had the same time. So this means that the percept is as one big parallel task that we have to work at. Yes, you had a question?
Yes, if you extend the x-axis outward, so I imagine if you have a super huge matrix,
yeah,
I imagine that it might take you more time. In other words, if you only have 25, that's that, right? It's a 5 by 5. But if you have 10 by 10, maybe it will be, like, I don't know if you did experiments with more, but I think that that's not.
I think these are considerations of, of the visual field and things like that. If you make it humongous and now you have to move your eyes and things like that, or move your head, I don't know, to cover the whole task, then other things come into play. But my point is to compare these 25 to these 25 to these 25, the same kind of matrix, and this has important implications on the task at hand. Okay, so and if you start to do different things, yes, probably different strategies are needed to discover it. But if she just makes the task harder by having the cross intermingled, for example, with different similar colors and structures that one has to identify within this more complex matrix, you see that the higher the number of items in the array in this matrix, then it takes more time for the subject to find the cross. And what she concluded from this is that there is a completely different task, of a perceptual task. Here it's a parallel task where everything pops out, and here it's actually a serial task where one has to search with his eyes the whole matrix. And to prove that is that when she asks if the item is absent, okay, in order to give an answer whether it's absent, you have to, by definition, you have to go through all the matrix. This will take you double the time. And in fact, every time the item is absent, it takes double the time. So this linear relationship and the double time between the different tasks got her to conclude that actually it's a completely different task of the visual system to identify the blue cross versus this task. Now this has important implications of how we encode, of how does the brain encode sensory information. And this actually led to a hypothesis. One of the most important one actually came from the Hebrew University, PhD work of Merav Achisar and Shaul Hochstein, where if you look at the visual system, you see that early on, and we will talk about that in more detail, after the retina, LGN, visual cortex, you find neurons that are responsive to these types of stimuli, okay, the orientation of the lines, for example. And as you go higher in the hierarchy, you get more and more complex cells until you get to the Simpson cell I showed you last time, okay? So if you stick an electrode in the retina, you will not find the Simpson cell. If you stick an electrode in the area, I don't know, IT, I don't remember where it was exactly, it doesn't matter, then you can find more and more complex categories of vision that one has to perceive. Great. And when they did this kind of quantifying perceptions, they actually found that it's much easier for us, or faster for us. We can identify objects much more rapidly than we can identify, what is the orientation of a given line in a matrix of lines. So if I give you, I don't know, many faces, or things of the type that Ant Risman did, and Shaul will probably talk about this in detail, and this gave rise to the what they call the reverse hierarchy theory of Merav and Shaul. And again, he will talk about it in detail. I just want to give you a flavor of the types of things that people do. And in fact, that the first thing that is happening perceptually is happening in higher brain regions. So if you follow, normally we follow the neural trace, what's happening in the retina, what's happening in the next station, in the next station, the next station. But in fact, if you quantify perception, it's easier and faster to identify complex objects, which have representation deeper in the brain. And what they hypothesized or put forward is a theory that says that, first of all, in time, the brain constructs categories and perceives that, and only later in time there's feedback connections to figure out the details. So it's actually easier to do complex things which in the neuronal hierarchy, in the brain hierarchy, are deeper down the line, okay? They're downstream. Excuse me? No, I'm just saying, again, Shaul will talk about this in detail. I'm just saying about quantifying perceptions, how these very simple things, even without sticking your electrodes and looking at the neurons, you can already think of, what is the problem that the nervous system has to solve. And these kinds of representation actually came from the neural recordings. Okay, so if you stick an electrode in LGN, you will see neurons with certain characteristics. If you put it in V1, you can see now orientation preferences. You stick it in V2, you can see, other things, until you get to Simpson cells and Jennifer Aniston cells. Okay, so quantifying perceptions is extremely important. And of course, this works hand in hand with what came actually later in history, which is the sensory physiology part. And the sensory physiology, when I say sensory physiology, I mean the relationship between the stimulus and how it is processed by the neurons of the brain. Okay? So these two subjects go hand in hand, and again, they become very powerful when we can quantify perception seriously and do the sensory physiology seriously. Only then you can see we will start to get a better understanding of how the nervous system actually encodes this information. Okay. A very dominant or common way to study the brain is to go bottom up, what we call bottom up, okay? Is to follow the sensory information. We first have to understand what's out there. So we need good microphones, good camera, good sensors to understand exactly what we're playing in the brain, what he has to perceive. And then if we're brave enough, we can go to the sensory organs and start to see how they respond to these stimuli in the world. Okay, and the question is, what do you think is important to know, and in what detail? Doesn't mean that these questions have clear answers, but just I want you to think, what kind of information would you like to know now you are entering the era of your scientific career? You have to be you are the scientist of tomorrow, and hopefully, I mean, you can find yourself doing, pure mathematics and computations, which is fine. But if you do, the question is, what do we really need in order to understand a system, a brain region? Let's say, the retina. Let's start easy. Okay, just take a brain structure. What kind of information are you interested to know until you think you solved the problem and we can move on to the next station of the brain? Again, until we reach perception, but that's a little far away. Yeah.
What properties of the electromagnetic radiation of the retina exactly perceive, or
Okay. So I would call this more input-output functions. Okay, what you put in and what you get out. So if you record from a neuron and you play 1,000 nanometer light in the eye of a human, you'll get a flat response. So input-output functions, I think it's perhaps one of the most important and basic things that one wants to know before he approaches a system. No, generally speaking. Generally in neuroscience. Okay, we have a black box and we want to understand how does this brain region work. When do you think you solved it completely? And we still did not solve it completely. Okay, that's a big question. Yes, yes. I'm trying to look for the ingredients. Okay, so the input. What's the input? I know in wavelength, pressure waves in the audition. What chemicals and what's the output? And then smart people like you can tell, calculate some kind of function, the input-output function. Yeah, shoot.
You need to know the anatomy of the connectivity.
Okay, so that's going a little bit more into mechanism. That's great. That's very trendy today, but it's generally true and very important because we can say, okay, the input-output function of this box is such, but now we have many neurons here. Okay, so I think something like, what's your name? Yoav said, we want, I think, if I can, extract what you said, a code in the nervous system is not done by a single neuron, but rather by a population of neurons. So we need to know the interactions between the neurons in order to really understand the nuts and bolts of the mechanism. So the anatomy is very important. Who is connected to who? Maybe, there's an all-to-all connections. Maybe only pairs are connected. Whatnot. We will talk about this also along the way. What else? So I know all the connectivity, and I know the input-output function of single neurons. Yeah. Great. So who these guys are projecting to? That's also very good. And why is that important in neuroscience? Because, and you will see this very often, people are, especially when they will look deeper into the nervous system, they're pretty much blind to who is the decoder. There is an encoder, and these guys encode information. But the part of the nervous system that has to read this code is the next station up. So when we say anatomy, it's not only local anatomy, who is connected to who, but it's very important to know, maybe only these two neurons send their output, their axons to another place, and therefore all this, population activity that we're calculating is irrelevant because the decoder doesn't have access to all the neurons in the brain. Okay? Of course, yeah.
so suppose the assumption that there's only one input in a sensory path, and actually each sensory path is affected by the other one and by the internal properties of the brain, like your memories and your.
Okay, fantastic. We'll get to that. You're a little bit ahead of me. But we're going to start slow. So there are many. Great, it's fine. There's interactions between different brain regions. It's not so much between, the retina is quite low in the hierarchy. But very early on, you start to see feedback projections and things like that, which also has to do something with the, with the anatomy, so to speak. There's information about, how these neurons work as a whole. And there are many other important things, okay? Because you need to know, that this neuron is an inhibitory neuron. This neuron can be an excitatory neuron. This neuron can be a low threshold neuron, and this is a high threshold neuron, et cetera, et cetera. So things that we want to know, or everything was raised up here. That's great. Your intuition works fine. Absolutely good. Inputs, output, the physiology of the cells, the constraints of the cells, how high it can fire. This is a lot of biophysics come into play here. The anatomy, the synaptic connectivity, the different cell types, which basically means, the strength, the sign of the synapses. And this is what Omri talked about or mentioned, the who-when combination, okay? The same neurons can be physiologically very different when the animal is attended or when it is not attended when you play a stimulus, Things of that sort are very important, yeah. Yes, so we will talk about the constraints. I would say the dynamics, maybe. Okay, so a neuron can fire only so much, and there are aspects of synaptic depression and facilitation. And when this neuron fires a lot, this neuron is shut down. And yeah, so you're talking again a little bit higher in the perceptual hierarchy. We're still quite low. And I assume you're talking about things like, in order to perceive something, I have to learn it initially and do the comparison to something that has been learned. These things are even less understood. I mean, absolutely yes, but we don't really know where they are in the brain. It's very hard for us to put the electrode or the neurons that will show us these kinds of complex or higher order interactions, perhaps if you will, but definitely we will not fully understand perception until we can have a good grasp on that, yes. So it depends on what you're talking about plasticity. If you're talking about basic processes like adaptation and things like that, definitely. I wouldn't think that interesting plastic events in the context of learning are happening at the receptors of the skin or the photoreceptors of the eye or the cochlea. But once you go one step into the nervous system, you start to see plasticity. Actually, I am myself, my own lab is working on relatively low or early parts in the nervous system where I try to claim that there is a lot of plasticity. So you're talking to the right person, but other people will, have different agendas. It's normally considered that, once you go into the cortex, then interesting things start to happen in the context of plasticity and learning, etc. So the history of the system is extremely important, but I think this is also a very good discussion or argument, okay? I won't say names, okay, because we don't want this to be a gossip lab. But, many people tell me an argument, who cares about development of the nervous system? It's not, it has nothing to do with function. All I want to know is, I have this nervous system, and this is how it functions. And what I need to know is the input-output functions, synapses, strength, anatomy, blah, blah, blah. Who cares what it was? It doesn't tell me, what happened during development. It may tell me how it got there. But I don't care if you got to Jerusalem from Haifa or from Eilat. You're in Jerusalem, and this is what I care. So development is important on its own right because, for example, for biomedical reasons, okay, there are developmental diseases that we want to understand and solve as a scientist, and there's no argument about that. Nobody argues that it's important to understand development for this reason. Is it important to understand brain function? I think yes, because we can learn. So other people think no, and there's a lot of fights between us. Okay, who cares? I think that there are interesting things during development that have very strong parallels to issues like plasticity and learning in the adult brain. Because the brain is very unique in the sense that it's continuously changing. You're always on a slope. So development is a little bit crazy slope, okay? It's very intense and steep slope because things are changing, all the time. And there are, very interesting things. Natan is sitting here from my lab, interested, I don't know, in infant amnesia. Okay? If you talk to a small kid, then he doesn't remember, or you don't remember what happened when you were 1 year old, and you had more neurons, and everything was changing all the time in your brain. So that's why, but maybe at some age during development, you start to remember, memories are starting to form, etc. And it will be interesting, I think, to understand the learning brain or the developing brain in this context. For example, of learning and memory, I think it's a very valid topic to study. But not all people think this way. Yeah.
In general about development, the brain is very complex and very hard to study. So if you take something that started very simple and then became complex, like you said, with the flow, so it'll be easier to start where it's simple and then.
Yeah, I don't think it's simpler. I actually think what we see is on the other hand, so that it's more dynamic. You have more neurons, more synaptic connections. They are more dynamic. They're changing all the time. And it's, you blink your eye for a minute and the picture is changing. So it's a completely dynamic system during development. So I'm not sure it's easier to study. In that sense, it's simpler to study. I would disagree with that intuition. I just want to take you a little bit to regular biology, if you will. Excuse my language. Okay, I come from biology. But if we look at development or genetics or cell biology, in fact, we can learn as neuroscientists a lot on how to do experiments, on what type of things we are interested in to study, because, frankly, they really understand the system much, much, much better than we as neuroscientists understand the system. And the way that geneticists look at or do the types of information, first of all, they identify the building blocks, for example, the gene or the proteins in the cell, whatever. When they talk about information processing, they talk about protein interactions, transcription, translation, degradation, blah, blah, blah. Again, not fully understood. That's why we still have many labs in genetics and cell biology, but they are much further down in their understanding of the system than we are. Okay, it's very nice. We can raise a lot of money for a new building, and the brain is the next frontier. But if you dig down deep, we understand the heart much, much, much better than we understand the brain, and we understand genes and cell biology much better. So maybe we should be a little bit humble and study, how they do experiments. And when they do experiments, they do things like gain of function and loss of function experiments, and test necessity and sufficiency. And you start to see these things in the nervous system as well. So if you look for parallels in neuronal circuits, so okay, we really now completely agree that the basic building block of computations in the brain is either a single neuron. If you look at the low animals, worms or whatever, or invertebrates, single neurons are the powerful building block that you can say something intelligent about what it is trying to compute. If you go higher, starting to get fishy and fuzzy, so hundreds of neurons maybe are a computational or building block, things like that. If we talk about information processing, which is what you guys are doing as the computational people, is looking about spike trains, adaptation, the computation, what kind of functions these neuronal circuits do. And people like me who do experiments are trying to use tools, and you will see this along the course, pharmacology, lesions. The new trends are optogenetics, which we can really manipulate the system at these time scales. molecular manipulations, genetic manipulations, but eventually we want to get to where the geneticists and the cell biologists are in understanding the system well. Now, if you look at neuroscience, and the sensory physiology, I think, is at perhaps the forefront of how we understand systems. Motor systems perhaps lag behind, and now, like I told you last time, there's a very strong interaction between the sensory and the motor communities because we think they should be one, should be united as one. Even when we look with our eyes, we are continuously giving motor commands to our pupils to move. And if you look generally, so the sensory information, sensory physiology. I mean, I'm sad to break you the sad news, but or good news, but this is where we stand. If you look at receptors, receptor cells, very early on in the system, this is best understood. We are really in a fair position, okay? How we understand the cochlea, retina, how receptor cells in our skins work. It's not too bad. Again, it's much simpler to study it. But once we go into the brain, the information flows in serially and in parallel streams, and along the way, the information is continuously transformed. It's changed. Sensory pathways, like people mentioned here, are recursive. You get a lot of feedback loops, and things go again in parallel, etc. And this is far, far less understood. So in that sense, you are coming in the right time to neuroscience. Definitely, things are not going to be solved by the time you retire. So you should be happy that you have a job and no problem. The brain is not a syncytium. This is actually a slide from my physiology paper. Okay, it's not about the number of cells that is important because, there are more kidney cells and, muscle cells, myocytes. But these guys are pretty stupid. They do generally the same thing. If you understood one myocyte, you understood them all. Things are great in physiology. The big trend, those of you who come from physics probably don't know, the biologists know it too well. Okay, there are great, different cells like the epithelial cells. Epithelium is very trendy. Because it has a membrane with completely different parameters. Okay, on one side of the membrane, there are different channels. On the other side, different things, and this has to coordinate transfer of information from one side to another. But again, you look at the epithelium, they're all the same. I mean, I don't want to insult this community, but they do the same. The real challenge in the brain is that neurons are different. Many of them are different. And each one is doing something different. So, the level of complexity is extremely high and gets very high very fast. So that's why we're still stuck. Okay? And again, I think we should be humble and learn from the people who do real biology because they really understand the system. And we're only blah, blah, blah. Okay? Not only, but to a large extent, we have really a large way to go. Okay. So, like I said, the first part of the Kandel paper, the Kandel textbook is basically handling or talking about things we do understand, which is low-level sensory neuroscience. And we're going to start at the beginning. The receptors is the first biological interface with the physical world. And if you look at different sensory systems, they have different sensory organs. These are called sensory epithelia. In vision, it's the retina. You'll talk about that. In audition, it's the basilar membrane or organ of Corti. We'll talk about that later on. In touch, it's the skin. In smell, it's the olfactory epithelium in the nose. I think we will not have time to talk about the tongue, although there have been great progress in understanding the taste system. Very cool. And maybe we will get to that if I have time, but I'm not sure. Okay, so, and different sensory systems have completely different designs. And the design is extremely important when you start to think of how what exactly is coded, etc. So these are the three, systems. For example, that I was not talking about the olfaction, but in hearing, it's a one-dimensional array of hair cells in the organ of Corti. In the retina, it's now a two-dimensional matrix of photoreceptors, somatosensation, also a two-dimensional spread out of the receptors on the surface of the body. The first thing you have to realize is the first transformation is from the physical variable to electricity. The language of the brain is electricity. Therefore, the physical variables, they change, and they come into play as electrical potentials, and these are called receptor potentials. So the first thing is a transformation of the physical energy of the different energies into an electrical one, which is the language of the brain. Okay, so that's the first transformation. We call this sensory transduction, and you have to realize that this is not physics. So it's not one energy which is changed into another one directly. Okay, because if we say a neuron responds to whatever energy with a spike or whatever, some kind of synaptic potential, so they are not equal. We have to pay. Okay, when you do signal transduction, you pay in biology, in money of biology, and the money of biology is called ATP, and it is metabolic energy. What? Yeah, fine, but it's not equal. It's fine. So look at this example. Okay, so one photon, and we can detect one photon, and it will cause a membrane synaptic potential of a few millivolts. Okay, now one photon energy is about 10 to the minus 18 joules. Okay, and in order to get that few millivolts, okay, you need to pay in flux of 10 to the 4 ions through the membrane. Okay, so it's not that this one photon that caused this is equal to it's not equal to the same energy, because in order to move 10 to the 4th ions from one side to the other side of the membrane, you have to pay in creating the electrochemical gradient, okay, across the membrane, which is something you all know from basic physiology. And each work of the pump of the sodium potassium pump, which gives us an electromagnetic effect of one ion, is caused, you have to pay 1 ATP, which means that this transduction is going through a metabolic energy step. So one photon will cause this depolarization, but you are paying you're paying basically the price of metabolic energy. That's the whole idea. Okay, so it's not a direct transformation; it's a transformation through the biological system, and basically this is where physics and biology interact, where this transformation is occurring. Great. Okay, so that's important, and this is called stimulus transduction, and the energy source is metabolic that we pay, so to speak, in biology. And the first interaction is where physics and biology interact, and once you go into the system, it's basically a similar language. Okay, and all different energy, be it light, sound, touch, etc., are transduced into electrical information, which is again the energy that we start to speak about is then metabolic from this point on, once it reaches the nervous system. And the things that cause the change can be very different. Okay, the mechanisms are different for this signal transduction, for this transformation from the different types of energies into electrical potentials. This can be mechanical coupling. Okay, this can be ligand-gated channels, like in the chemical senses. It can be second messengers cascade, like in vision and smell, and we'll talk about these as we go along. Okay, there's a table in the textbook that shows, what's the modality, what's the type of energy, light, sound, gravity, whatever, mechanical, specific chemicals, blah, 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 and what type of receptor classes. We have mechanoreceptors, photoreceptors, thermoreceptors, chemoreceptors, etc., etc. Each one is represented on a different place in the body, comes in different flavors and form, but all of them do roughly the same thing, which transform or transduce the energy from the outside world into the language of the brain. Okay, how are we with time? We're fine. Great. So these are called signal transduction, and the change in electrical activity in the receptors is called receptor potentials, and these are then generated in order to communicate between different parts of the brain by spikes. Some receptors themselves are spiky, okay, like in somatosensation or in smell. So the receptor which actually interacts with the physical energy gives a spike, and some receptors are not spiking, and only they will carry the information to the next neuron down, normally with a synapse, and only they will be spiking, okay? And spikes come into play normally when neurons have axons that they want to convey the information far away, but also there are neurons with no axons and things like that, but I'm not sure we'll get to those neurons. Yeah?
Is there a evolutionary incentive for one of those transduction methods?
Evolutionary what again? I have an answer, but I'm not sure I will answer your...
Is there one way which is more helping the evolutionary, and which is more I forgot the word for optimal for the evolutionary process, because I think the shorter the gap between the information from the physical world to electricity, it's better than the downstream method, which takes longer time.
So I'm not sure I know how to understand this. Okay, if you look at lower animals, let's say you look at C. elegans, okay? These animals don't have spikes to a large extent. They do have electrical potentials. Electrical potentials are something which is very common not only in nerve cells, okay? Many other cells have membrane potentials and things like that. And if you look early in evolution, the synapse was developed earlier on, okay, to start transferring information between cells. And only as the nervous system became more and more complex and long and physically long, and you had to transfer information efficiently from one way to another, you started to see these the spikes, for example, and axons and things like that. So I don't know exactly if I'm answering your questions and what is the right way, but the synapse was born first. That's for sure. Yeah.
The definition of a spike generation as opposed to not spike generation, is whether it's a linear transformation of the signal or a nonlinear transformation. Is that still trying to understand what the...
What is a spike? What do you mean? I don't know.
In other words, in vision, so you have a photon, it's your photoreceptor, so the photoreceptor does respond electrically, and that is what...
It's not a spike, not by a spike, though.
So I guess I'm trying to understand the definition of a spike, because it isn't you do have an electrical signal.
Yeah.
So what is it that you don't have? You don't have the potassium.
You don't have the classical sodium spike with a threshold. It's a continuous signal. Actually, In vision, you will see that it's the opposite. It's a depolarizing, it's a hyperpolarizing effect, but we will learn about that. A spike is an all or none event that is triggered when you cross a threshold, and you will learn lots about it starting Sunday. Okay, Dan will be here, and he will talk about dendritic computations and spikes, etc. I'm just saying that, and normally it's when I say it here, it's spikes normally travel along an axon. Again, I have to take back what I'm saying immediately because there are other exceptions, but this is a very efficient way to get information in bits of information, if you will, electrical potentials which are not analog, if you will. Okay, and these go through spikes to downstream regions. Okay, let's take a break, 15-minute break, and talk about some of the boundary conditions we have in sensory systems. And then, if we have time, we will talk a little bit about tips for representations. I know some of you are from, I don't know, from psychobiology, and the stage is home for them, but some people don't. So, so from previous years, people asked me to give them pointers to what is important and what not, and we can do that if we have some time left. So let's take 15 and be back in 15 minutes. יותר מהירה, כן, אבל היא לא יותר יעילה כי יש לך פחות רמות של שליטה. אתה משיג, לשנות, לעשות מניפולציה. יש כל מיני, בעיקרון זה הכל ביוכימיה, הכל זה מולקולות וחומרים שמפריצים, ויש להם אימפקט על החשמל, יכול להיות. Yeah, we are very close to that. Every year we have something. And yeah, so yeah, I think it's the best. Just come an appointment and explain to exactly what you want to do. We set some kind of date. We have 2 days of seminar. Once in your semester, you are obligated to do one semester. This is the semester. We have 20 students, so we have to do 50 meetings, and then we have to do 20 easy. Yeah. Just write me an email. I'm just flying tomorrow out, so I'm not going to be in the next 2 weeks. Where did you? I remember you. You were. Yeah, and did you find the lab? Udi. Okay, great. And everything is okay? You found a place to live and all the bureaucracy?
במקור זה הכוח, או VST.
first of all, I lost my battery, so you have to live with my voice. And we continue with the very, very, very basic, the ABC of sensory systems and physiology, okay? Great. So first of all, that we should realize that neurons are basically spatial and temporal filters. They filter the information, and they have spatial properties and temporal properties that are important for their function. Okay. This is, for example, neurons, or every neuron, or many neurons in sensory systems are characterized by concepts like, or words like tuning width, best frequencies, receptive fields of the cell. Okay? And these are basic features that when we study sensory systems we normally refer to. So here, for example, you see some classical cells in the visual system. And for example, you have three types of cones responsive to different colors. The red cone, for example, would have this kind of tuning curve, peak at 564 nanometers. The green at 533, etc., etc. Rods are more in the center range, around 500 nanometers sensitivity. But this is a classical tuning curve of a neuron. Okay? And when we look at these tuning curves, you will see measures like tuning width. Okay, what is the width of this tuning curve? Some neurons will have narrow tuning width, some will have broad tuning width, some will have one base frequency, sometimes neurons with two peak frequencies, things of that sort. Yeah.
What's the y-axis?
Here the y-axis is the number of spikes, for example. Okay, the physiological response to this stimulus in this case. And if
But these are photoreceptors, right? They're not supposed to have spikes.
it doesn't matter, membrane potential hyperpolarization in this case. Okay.
Okay.
You're right. Okay, it doesn't matter. The physiological response. Okay, so if this is a photoreceptor, he will respond more strongly at, or at the peak response to these types of. Now, of course, there are, if you're only looking at the red cone, in this case, and he gives us hyperpolarization of whatever, 10 millivolts, we don't know if it's 500 nanometers or is it 620 nanometers. When you look at only a neuron, and of course, the solution is to look at many neurons simultaneously. So a single neuron normally does not give you enough information because of these structures that you're tuning with, because they're not, in many cases, they are not perfect feature detectors, certainly not when you look at basic physical measurements. Okay, so they have some kind of a tuning curve. Here is an example, and if you want to convey the information, you want to know exactly what is the precise color. For example, you will need to look at more than one type of cell. Okay, and do the cross between them and the intersection between them. And only then, as a decoder, you will know exactly what is the frequency. It's the wavelength in this case. Okay. Many cases they are irregular, so I just showed you a putative, perfect tuning curve. This is a real neuron from our lab in auditory cortex, for example. And neurons have come in very, very different flavors. Okay? This neuron, for example, what you're seeing here, again, additional terms that you need to know in neuroscience. This is what we call a raster plot. A raster plot is when you plot the spikes along time, normally with different repetitions to different stimuli. In this case, this is just different tones, okay, from 3 to 80 kilohertz each bar here is 20 repetitions, 10 repetitions, whatever, okay? And what you see here are the spikes that were the responses of the neurons and spikes to the different repetitions of the different stimulus. So in this case, there are, many, many repetitions for each stimulus, in this case at one intensity, and this is the type of response that you get. So you can see a few things. One, this neuron is responsive. Okay, the stimulus is given somewhere here, let's say, in this peak, okay, for, I don't know, 50 milliseconds, it doesn't really matter, the time, okay, so there's a short stimulus. You see this neuron is rather sparse in its activity, not firing a lot. Nevertheless, it does have a spontaneous firing. So the spontaneous firing rate is not zero, it's a little bit over zero. And you can see spikes, here and there, which are not coupled to the stimulus itself, which is given here. You can also see that this neuron has some kind of tuning width. It responds to frequencies between, I don't know, whatever this is, 10 kilohertz and whatever, 30 kilohertz. This is a logarithmic scale, but never mind. But you can see that it responds not to the whole spectrum of sounds that we gave it, only to a given spectrum. Okay? And maybe the responses are stronger here, etc. Another thing that we use, and you will see a lot during the course, is when we collapse this image into one dimension. This is called the PSTH, post-stimulus time histogram. And basically it reflects what we see here. This is we're losing the information of all the frequencies. Okay, if we want to look at the frequencies, we would have to do another reductional dimension on this axis, and then you will see exactly what frequencies this neuron responds to. Okay, but the PSTH is something very, very common to see the locking of a neuron to many stimuli. So this is called the PSTH, this is called the raster plot. Yeah.
So the raster plot, do you present like different drives for different frequencies? They are presented at different times, at the same clock?
They're presented at different times. So each line here, there are many, this is constructed of many, many repetitions. Okay, so let's say we have 10 repetitions. You play the sound once and you get a spike here. And you play this, so this is the first repetition. You play the sound again, you get 2 spikes. You play the sound again, you get no spikes. You get to play the sound again, you get no spikes. Sound again, no spikes, again, 5 spikes, 1 spike, and the stimulus is here, and no spike, I don't know, 2, 4, 6, 8, 9, 10 repetitions. So this is a raster plot. No, same frequency, 10 repetitions. Okay? So what you see here is actually, I gave 10 repetitions for each frequency. So this was for the 80 kilohertz. And now we have for the 64 kilohertz, whatever. Okay?
This is like a zoom in to 1 bar.
This is zoom into 1 bar with 10 repetitions, and each tick is a spike.
Okay, and
So there's a lot of information. Yeah.
What can we learn from the PSTH plot? Because it's like besides the latency of the very average response.
So you can look at the latency of the response, for example. Okay? Or the intensity of the response in terms of the total number of spikes.
So but it average over different frequencies in this case.
Yes, and so you lose. Once you reduce the dimension, you lose information. Spontaneous firing rate is also something that we learn about. Okay, so you see here the spontaneous firing rate is as such. If you look at inhibitory neurons, so if this is the classical PSTH in the auditory cortex for that's the stimulus for a pyramidal neuron, I don't know, in layer 2 or whatever. If you look at an inhibitory neuron, you will have a much higher spontaneous firing rate. It will respond later, like you said, latency. Maybe the duration of the response is different, so you can also learn something about the duration, things like that. This is the normal types of things that we learn, not only latency to respond, the duration of the response, the number of spikes, again, not per your peak stimulus, basic characteristic of a neuron. Yes.
Is the same neuron or the same type of neuron? Or
This is, for example, classical for a different type of neuron. So this is only
neuron?
what?
Is it really the same neuron you do this?
This? Yeah, the raster plot here is the same neuron where you play many stimuli. Okay? If you take a different neuron and you do the same experiment, you will get this type of PSTH, for example. Okay? Neurons will be very noisy. For example, we still don't know the contribution of spontaneous firing rate. Okay? Some people, that's their bread and butter. They look at signal-to-noise ratio, or what's the basic spontaneous firing of a neuron, and this can change in different aspects. For example, an animal is attending to a stimulus, and you can see a spontaneous firing rate change. What does it mean? You see a reduction in the signal-to-noise, in the population level, etc. Welcome to noise. In the auditory system particularly, we also plot things like frequency response areas. Excuse me, Adi, which is doing this for a living, probably. Okay? So now, if you look at the receptor field in the auditory system, we build something like this. So this is what you saw before. Okay? That's the PSTH, that's the raster plot for a given intensity, okay, which is the highest intensity. In this case, 72 dBSPS. And then you can collapse this raster plot even to a color diagram like this. And what we do next is we play the sounds at lower intensity. Okay? So if you look at the raster plot very well, and this is when we play it at lower intensities, and that's the PSTH on the top. I'll go back. Okay, so that was the PSTH initially at high intensities, and now at lower intensities, and now at lower intensities. Now at lower intensities, you're basically left only with the spontaneous firing rate because the neuron is not responding anymore. The PSTH is flat. And we get these kinds of tuning curves, which we call frequency response areas. This is in the auditory system. But for each sensory system, you will have a different type of receptor field that people see. Okay? And this gives you, the highest sensitivity of this neuron is actually here, because sometimes they're not perfect, it's not like a band. We call them V-shaped receptor fields because they have a V shape in many cases. So they're broad towards the lower frequencies. When we learn about the auditory system, we will know why or learn why. And as you go lower and lower in the intensity, they become sensitive in a narrow range, and people can talk now about calculate thresholds, tuning with size, sensitivities, all kinds of measures. And this can change with plasticity, with learning, with attention, you name it. Okay? And again, one neuron, many neurons, where these neurons are sending this information. Again, welcome to noise. And why is this here? Yeah, anyways, I said this before. To code the informative information you need from population of cells, not from a single cell. You have to remember that the receptor field is a very basic, the raster plot, tuning width, etc. is a very basic description of the receptor field, but still it is very limited in how much we can extract information with it. And this is for another example from our laboratory, work of Ami Shalev in collaboration with Eli Nelkin, looking at 15 different neurons. And just for the sake of argument, okay, I don't want to start to go into the data, let's say that all these neurons have roughly the same receptor field, or frequency response area, roughly. And if you look at neurons, even though they have the same receptor field, but how they respond not to pure tones, because when I talked in these slides, it was just very simple, stupid, if you will, pure tones, beats. Beep, 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 beep. We play all these sounds and construct these diagrams. But in nature, animals have to respond to complex stimuli. And this is true generally, okay? If you look at an image, or if you hear a sound, or music, or whatever, it's composed of long sequences of energies, like this one. This is in particular localization on the top. Omri is now the master of localization. If you have any questions to Omri. But these are recordings from neurons in the, I'm revealing all the secrets. And these are recordings from neurons in the auditory cortex. And you can see some neurons respond only to the beginning of the sound, which is basically noise. Some neurons respond to each and every syllable in this call, okay, this pop call. Some respond by 100% probability with 2 spikes. Some respond only to the first syllable. Some respond very strongly to the 7th syllable. Some respond on the top there, they're quiet. I mean, it took us, I don't know, 30 minutes to get the information from each raster plot here. Okay? Where's my... Oops, I lost my... here it is. Okay, look at the top neuron, neuron number 1. Responding very, very sparsely, only to this syllable and this syllable. And all the rest is completely quiet. The spontaneous power rate is 0, and it responds to these syllables with, I don't know, 20% probability. So I played the sound 10 times, he responded twice or 3 times. Whatever. But again, so you see there is a very complex task to solve for neuroscientists. Because the picture is much, much more complex even from a basic receptive field, which is again something that you will encounter again and again in the course. Okay. In terms of techniques, we must mention the giant. Vernon Mountcastle died last week, 2 weeks ago. He was the first who started with looking at sensory physiology in cortical structures, and these are Hubel and Wiesel, which followed the footsteps of Mountcastle, or Vernon Mountcastle, and he did experiments in the somatosensory system. They started doing experiments in the visual system, and this is classical stuff. And on our series of classical papers, you will be requested to look at their papers. It will be part of our exam. But basically speaking, just to give you a feeling of how they did their experiment, so they're recording in the cat visual cortex. I don't know if you've ever seen this. And they're playing these bars on the screen. Okay, and then they take a niyar shakuf, I don't know. They put it on the screen, and they start with a marker to paint, where on the visual field of this. Okay, so they sit there with a cat looking on the screen, and this is the way they start to plot the simple cell 1962. Okay, it's a 12-minute movie, okay? So let's not... and they're, anyway, they're plotting receptive fields. Great. Anyway, this is different neuron. Anyway, classic stuff. Complex cells, etc. We will go back to it when we talk about okay. That's one thing, and 2 more modern stuff. Last week in Jerusalem was Dan Dombek. Sorry. No, really no agenda. Trust me. He's beautiful, Yair Lapid, but not my note. Anyway, so what you see now here is mice, head restrained mice, running on a ball in virtual reality experiment. And this is now the gold standard. Normally people either record from these mice or do imaging, so you can put a microscope above the head. This guy's running, left and right, and doing full tasks, so they can run in corridors and get their types of things. The virtual reality experiments are now things that we can do in mice and go deeply both into the psychophysics, if you will, to see when do they make decisions and things of that sort. Whoa. So, great. What else? What else? There's another video. Yeah, okay, so let's not spare you the video. Okay, there are different types of measurements that people do. In neuroscience, in 1950s, Bert Sackman and Erwin Wiener invented the patch clamp technique so they can record from single channels. You will see experiments where we record with electrodes, spikes, currents. If you patch clamp the cell, I don't know if you learned about voltage clamp, okay, great. Current clamp modes, spike trains, and there are other ways of measurement. So one can stick an electrode and record the local field potential, which picks up information from large populations of cells, and even put electrodes on the head of the animal. Okay, the big advantage of electrical recording is that this is the real language, so to speak, of neurons, superb signal-to-noise ratio, and superb temporal resolution. Now there are other methods that complement electrophysiological recording. I apologize for those of you who've seen this. This is the two-photon image of brain flooded with calcium indicators, and basically we're focusing our microscope at roughly a little bit under the dura mater, okay, of the brain, and this brain is filled with two dyes, one to label astrocytes and one to label neurons in green. And now we change the focus. Okay, we go down into the brain. This is a live mouse under the microscope. These are blood vessels. Okay, these things that go, and what you saw are the green neurons that are labeled. Okay, so let's go down again. These are blood vessels. Actually, the red glia, they cover the blood vessels. It's like when you heard about the blood-brain barrier, it's actually these cells. They have mosquitopodia that they hug the blood vessels and make this interface. And what you can also see is these shadows. Let's stop the movie. Where is my cursor? I think I made some kind of mistake. Here it is. Okay, so let's take the movie here, okay? So this is the heart of the injection, of our injection. Again, this is from my lab. And you can stop here, and you have, maybe 30, 40, 50 neurons that are labeled with the calcium indicator. But you can also see how many neurons there are in this brain region. This is the cortex, the auditory cortex. You see all these black holes? Those are neurons that were not stable. Okay, so we call them shadows. You go deep into the brain, you see all these shadows. So basically you have thousands and thousands of cells which are not recording, and essentially anytime you play a sound, thousands of cells will respond. So it's our responsibility to try to make sense out of this mess. What we can do now is record calcium transients, in this case from 16 cells simultaneously. So you focus on one thing, and you get these types of recordings, and now we have to translate each of these bumps into spikes. It's a disadvantage because it's not a direct measure of spiking activity. This is calcium, which is a great sensor on its own because its basal level is very, very low, and its correlation with spikes is rather good in some neurons. Yes.
Video, some of the shadows looks like they're moving. Is something actually moving, or is it
No, not moving. You're just moving in focus, and you see the big holes are the blood vessels. Okay, these are the blood vessels.
So the detector is moving while the video is
No. The whole the objective is moving.
the objective, yeah.
So your focus is moving. Yeah. So basically, we go down from 0 to minus 400 micrometers. So it's roughly down to layer 4 of the cortex. Roughly. But anyway, we stop in the middle, and you get these kinds of information. this is fantastic to computational science. Because now it's not just, first order information, or you can do correlations until you fall asleep. Okay? And people, computational scientists like to do correlations. Okay, anyway, I love correlations. Don't get me wrong. Even as a biologist, I have been affected by my immediate environment, and I'm also sinning. Anyway, these, you can find labs, a few labs in the world who are doing, have machines like that, basically building the microscope on top of the head of the animal. Even here, if you go to Eddie Nelken's lab, they have not microscopes, but electrical recordings that they implant on rats while they interact with the environment and things of that sort. So we are moving, the science is moving in this era into recording from awake, behaving animals that are engaged in the task while we record the neurons. And in fact, when you get information from the neurons, it's extremely important in some cases. Yes, some cases no. The exact behavioral situation of the animal during the task. Yes. So the state of the art, so there are arrays of electrodes, electrically or optically. Electrically, the house, it depends who you ask, okay, and the quality of the recording varies, but you can have arrays of up to 120 electrodes these days. So it's like a comb, array of combs of electrodes, frightening, quite frightening, that they put into the brain. To record electrically, you must penetrate physically into the brain. Some of these electrodes have silicon material on them that they can record in good quality. So there are arrays of 120 electrodes, for example. But normally, frankly, I do not see, don't see many papers with more than 10, 15 good isolated recordings from these electrodes. In optical imaging, you can get hundreds of cells. There are many, many new tricks to image in 3 dimensions. Okay, you can take objectives that jump and things like that, and you can get volumes. And I've seen, like the state of the art is thousands of cells simultaneously. People do now the fanciest thing is zebrafish and fish, so they can get all the brain, all the neurons simultaneously while the fish is flickering its tail. Because these animals are transparent and there are all kinds of, not a super high resolution, but there is a payoff. I mean, you have to pay a price when you go to optics, etc. Yeah.
What is the temporal resolution of the calcium imaging?
The calcium imaging? So it depends how many neurons you want to image. If you want to image a few cells, a handful of cells, you can go pretty high. You can go up to 1 kilohertz, but not many people do kilohertz imaging. If you do voltage sensitive guys, you go to gigahertz. But then you measure synaptic potential, not spike. Calcium imaging, I think, is more limited by the sensors themselves, which are slow, rather slow. So spike, what?
The GCAM?
There?
The GCAM?
GCAM?
Yeah.
Dye? Yeah, those are, those are just the genetically encoded sensors. The issue, they're faster, better, synthetic sensors. A neuron gives you a spike, okay, width, which is about, 1 millisecond. The calcium signal that you detect is then, it's a slow event. Slower to rise than the spike and slower to, of course, to decay. So calcium are, you don't need to go to 10 kilohertz, but you can go. I mean, it's not the limiting factor of the machine.
Decay of the chlorophyll or the decay of the calcium?
Decay of the calcium, yeah.
Oh.
Decay of the calcium. Yes. Chlorophyll works much faster.
Calcium is a very limiting factor in terms of actually realizing the correct pipeline.
Absolutely. So it's very useful if you look at
sparse neurons that spike every second or just beyond it. Once you start to have neurons that spike a lot, then it starts to, you start to get things like this, and there are ways around it. But anyway, calcium is an indirect measure to retinal activity, and if you're really serious about timing, and again, many computation people are very serious about timing. If it's puck puck or puck 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 or whatever, or puck puck puck, it's a whole difference. If the code is in the rate, or the number of spikes, etc., so it makes a difference. But if you want to look at large populations, genetically identified cells, things of that sort, imaging is very important. Okay. Sometimes, mostly not, as you saw in the wriggling call example, sometimes there is a nice linear relationship between the psychophysics and the physiology, like in the Simpson neuron. Or here, not exactly in the Simpson. Simpson was super cool, but we didn't have really an input-output function. Here, for example, if I ask you, you saw this before, how deep am I pushing your skin, you will have a rather good resolution on the intensity of skin indentation. And if you record from the cells, the photoreceptors, the mechanoreceptors on the skin, you also get a good linear relationship, which means that this is a good way to encode this measure. Okay, and you see, for example, neurons, when you learn about sensory system, some neurons respond like this. Okay, this is the intensity of the pressure. And as you increase the pressure, you increase the spike rate. And you can say that the number of spikes, instantaneous frequency of the spikes, is a good measure of the intensity of the stimulus. In other cases, you see different kinds of neurons. So for example, these are rapidly adapting neurons. They can spike rather fast, but then when there's no change in the measurement, there's no spiking anymore. Okay, so these neurons are actually coding changes in the intensity. You see this also in the auditory system. Okay, neurons which respond to frequency modulation. So not all neurons are the same. Different neurons have different flavors, and what you want to eventually understand when you study a sensory system in this case is what exactly is the neuron coding, what exactly the population codes, etc. Yeah.
Is the possibility to change the input, is it a single neuron characteristic or could it be a network characteristic?
So in this case, in the mechanosensory system, it's a single neuron that codes this information. But again, as a decoder, you have access to many neurons. There's more information than just a single neuron. But very early on, like I said 10 slides ago, we have a good understanding of what is out there in the world. What is the frequency, blah, 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 things of when we take the energy and break it into its classical bits. When we go into higher and higher, category of stimuli and things like that, you can't find it in the receptor itself. But things of this sort of change, you can even find at this level. And in general, if you look at humans, or depending on the animal, it's a population. If you look at an invertebrate, I don't know, a spider, or a worm, you can have information encoded in a single neuron. Okay? So for example, I did my PhD on cockroaches, which detect, they have on their, called Circe, it's like two antennas on the back, they have these hair cells, receptor hair cells, and they cover the whole space. So when you run with your sandal to hit them, or a frog sends the tongue to hit them in nature, they can detect exactly where they are in space and run to the other side. And these are done by single neurons. So single neurons cover a good percentage of space, and if this neuron fires, then the cockroach will run to the other side. some things are encoded by simple spatial neurons, and the lower you get, the simpler you get, the more common it is. Okay? Enjoying an orchestra, the opera, or whatever, you need more than 1 neuron. To enjoy, how she changes her voice, whatever, the thinker. Bon. Where are we? Fidelity to the physical stimulus. Okay, so there are different kinds of limitations that the neurons have to deal with. Like I told you, they are spatial and temporal filters. So when we say spatial filtering, we talk about sampling. And we talked about receptive fields. And if the more common thing is to know how much of the skin this neuron is covering. How many receptors do we have on the skin, or patch of skin? In vision, it's the angular extent that the photoreceptor has access to. In hearing, it's the range of frequencies. And generally, it is referred to the relevant dimension, which is encoded topographically on the sensory epithelium. In many cases, not all cases, you see a very nice topographically organized sense organ. Okay? So in vision, it's very intuitive. Map of the world is mapped on the retina. I think the axis of the retina, two-dimensional retina, has access to the world. So this photoreceptor has access to this part of the world. This photoreceptor next to it is mapped close to it and has an overlapping, but very close. So you can really make a map of the place of the cell and its responsiveness to the world. And in fact, in the visual system, this topographic map is carried on a few stations. When you get to the Simpson cell, already this map is completely lost. So different rules govern. If you look at the auditory system, very similar. You see that the cells are organized in what we call a tonotopic map. So they are a one-dimensional array, and if this neuron responds to, I don't know, 10 kilohertz, this neuron will not respond to 80 kilohertz, but to 12 kilohertz, 14, etc., whatever. Okay? They are nicely organized in a topographical manner.
Those are the neurons in the fovea?
Yes. Okay? And in fact, if you look one station higher into the brainstem, and a little higher, even in early cortex, you will see a very nice topographical map, which means that this information is probably a very basic feature of the system to extract information. In the chemical senses, it's a little bit of a mess. We have no order. All the cells are distributed randomly in the epithelium, and you will learn about that as well. Okay? But what we know most about is the other senses. We don't know much about olfaction and taste as we would want. Okay, so first of all, there's spatial filtering, and there's limits to the spatial resolution. Clearly, the more receptors we have, the more fine detail we can extract from the system. And the resolution of the system depends on the number of receptors. This is very obvious. The number of receptors and the receptor field, structure. This is, if you take an image and you put more receptors in, you can get a crisper, a higher resolution image. Now, the nervous system is built in such a way that the receptor density is different in different places. Okay, so your receptors on the back, you know this from high school, are very different than the receptors, mechanoreceptors, I don't know, on the lips or fingertips, especially have high density of receptors, so high resolution, and the location changes very dramatically. The visual system, for example, also, there's a region of high density of receptors, which is called the fovea, and the resolution of vision in the center field at the fovea is very high and is reduced very fast as we go away from the fovea. Okay? Yeah. Only spatial. Fair enough. Okay. Normally the initial step, wow, we don't have time, huh? Okay, so let me talk very fast. Normally, the initial step of processing is to break the stimulus into its components. So if you look in the cochlea, neurons are tuned to specific frequencies, which is the basic parameter that defines auditory space. Same thing in the retina. The neurons in the retina will break the information into the exact space and time, the wavelength, etc. And as you go higher into the nervous system, more and more complex features of the stimulus will pop up. How much receptivity overlap? This is basically a physics thing. So those of you from physics should know this much better than I do. If you want to reconstruct a stimulus in full, it's called the Nyquist criteria. If you want to reconstruct a stimulus, which is what we call bounded, which is a band-limited signal, you must sample it at least twice the rate of change, the maximum change of the stimulus. If you will not do that, you will have aliasing, and the signal will be different. So let's say this is a 5 kilohertz or megahertz sample, it doesn't matter. Let's say 5 megahertz sine wave, and if you sample it at 1, you can reconstruct this sample. If you sample a 5 megahertz sample at 5 megahertz, you will not be able to reconstruct it, and you will get aliasing in the sample. If you want to get a full reconstruction of the signal, you must go twice as much as the sample. Okay, that's a basic Nyquist criteria, which is known from again physics, and this means that we have some kind of limitation. So if our receptors in the sensory organ have a given dimension or density, this will be a limit of the system itself. Okay? So if you look at the So if you look at the visual system and you look at classic optics, each spot has some kind of coincidence function and will have a width. You can do the calculation at home. Eli did it for you. Here, basically, even a spot from a distance, because of the physical limitations of the eye, will fall on a 1 micrometer spot. So there are limitations on the sensor itself. And if you look at the density of the photoreceptors on the retina, they're spaced at 2 micrometers apart. So at best, we can reconstruct in full a 4 micrometer change in the dynamics of the visual system. Okay, so just to say that we have a spatial limitation on our sensors.